So, hello to all. I am Els Vergoeyen, the president of the French Society for Cell and Gene Therapy. Now, the board and myself are very happy to share with you a series of webinars on gene editing and gene therapy. I hope you saw the previous webinar by Juliette Rosier, who gave a very nice introductory speech on this. So otherwise, you can always go back on our via our website and our YouTube channel. Now, the board of the SFTCG really wants to give the opportunity to PhD students or postdoctoral students to present their work. And I am very happy today to invite Laura Ugalde for the next talk in our webinar series on gene editing and gene therapy. She graduated from the University of Francisco de Vitoria in biotechnology with a master's degree in innovative therapies. And she also got, as I understood, quite recently her PhD in November last year. So she's definitely a young scientist. And currently she's conducting her first year as a postdoc fellow at the Biomedica Innovation Unit at the CIEMAT in Madrid also. And this under the supervision of colleagues of mine with whom I'm working for a long time, Dr. Paula Rio and Juan Buren. And she will focus there on new gene editing strategies for the correction specifically of Fanconi Anemia hematopoietic stem cells. Now, she also got rewarded last year the prize for the best oral presentation. So I think we will have a really nice talk. Please, Laura, go ahead.
Okay, thanks, Els, for the really kind introduction. And also to all the board members for allowing me to be here and being part of this webinar series. That is a real pleasure. So today I will be talking about our latest data focus on using different gene editing tools to repair specific mutations that cause a devastating disease that is known as Fanconi anemia. So Fanconi anemia is a rare hereditary recessive disease that affects 1 in 136,000 births, which is caused by an impaired DNA damage response that leads to genomic instability and in the last stages to bone marrow failure and an increased cancer predisposition, which is the main cause of morbidity and mortality in these patients. So Fanconi anemia is caused by mutations in any of the 22 genes that participate in the DNA damage response FABRC pathway, from which the most frequently mutated gene is FANK, which has an estimated frequency of around 80% in the Spanish population. Mutations in these genes results in a cellular phenotype that is characterized by chromosomal instability, a late S and G2 cell cycle arrest, and also an increased sensitivity to reactive oxygen species and ICL-inducing agents such as acetaldehydes or mitomycin C. Then among the clinical manifestation, an increased sensitivity to pro-inflammatory cytokines that is associated with the pro-apoptotic phenotype involved in bone marrow failure. So among the clinical manifestations, approximately 75% of Fanconi anemia patients present congenital abnormalities, including skeletal defects, irregular skin pigmentations, and cardiac or neurological anomalies. Also, these patients have an increased cancer predisposition, especially myelodysplastic syndrome, acute myeloid leukemia, and solid tumors such as head and neck squamous cell carcinomas. Even so, the most common feature of the disease are the hematological defects that appear during the first decade of life due to a reduced number of hematopoietic stem cells, and which leads to pancytopenia and bone marrow failure. So in some patients, due to the chromosomal instability of Fanconi anemia cells, a second or even third spontaneous mutation can occur compensating the original one, allowing the restoration of the protein with a different amino acid composition but a complete functionality. This spontaneous reversion is known as somatic mosaicism, and when it takes place in a hematopoietic stem cell, its subsequent expansion to every cell of the hematopoietic lineage gives rise to the improvement of patients' peripheral blood cell counts, that in some cases stabilizes bone marrow failure progression, and which can be considered as a natural example of gene therapy. Even though nowadays the only curative treatment available for the hematological manifestations of Fanconi anemia is allogeneic hematopoietic stem cell transplantation. Even though its application is hampered due to the difficulties of finding a well-matched donor, also the hypersensitivity of Fanconi anemia patients to conditioning regimens, and the risks of graft versus host disease. So in this case, the correction of patients' hematopoietic stem and progenitor cells by gene therapy is a promising therapeutic alternative due to the proliferative advantage that is associated with the correction of mutated Fanconi anemia genes, which has been demonstrated for the first time in a clinical trial conducted in our laboratory, in which patients' hematopoietic stem cells are collected, then transduced ex vivo with a fungal antiviral vector, and then reintroduced into the patient. So in this context, the use of targeted nucleases to induce double-strand breaks in the genome for therapeutic purposes opened a new era of precision medicine, in whose resolution the non-homologous enjoining or the homology-directed repair strategies have been mainly involved. The non-homologous enjoining repair mechanism is the cell's first choice for double-strand break repair, in which machinery processes both ends in order to generate insertions or deletions that are directly ligated without the requirement of a donor template. This repair mechanism is the preferential system in quiescent cells, such as hematopoietic stem cells, and is a simple mechanism which only requires the nuclease delivery. Also, importantly, in the case of Fanconi anemia deficient cells, this repair mechanism is enhanced. On the other hand, the homology-directed repair strategy relies on the accurate homologous recombination repair pathway, which is only active during S and G2 phases. The system is more complex, which requires the use of a donor template and throughout its ability, and in contrast to the non-homologous enjoining, is partially hampered in Fanconi anemia. So taking this into account, we exploited the error-prone nature of the non-homologous enjoining repair pathway in order to generate insertions or deletions by CRISPR-Cas9, mimicking the natural reversions described in Fanconi anemia mosaic patients. So to study this, we selected the most prevalent mutation described in Fanconi anemia patients from Spain, which consists on a homozygous C to T substitution in exon 4 of FANK gene that generates a premature stop codon, leading to a truncated non-functional protein. So in order to target this challenging mutation, our strategy required the removal of the premature stop codon and the preservation of the open reading frame without disrupting any functional domain of the FANK protein. So in a first set of experiments, a mutant-specific single-guide RNA was designed based on the closest distance to the mutation in order to minimize the size of the indels required to remove this premature stop codon and then preserve the open reading frame of FANK sequence. So this single-guide RNA was tested in a patient-derived lymphoblastic cell line harboring the specified mutation, in which we observed that the generation of frame-restoring events was highly efficient, reaching up an 8.5% 5 days after electroporation, that not only removed the premature stop codon, but also preserved the open reading frame of FANK sequence. We also observed that these potentially therapeutic indels showed a marked enrichment over time when cells were maintained up to 60 days in culture, reaching up to 25% of potentially therapeutic indels that restore the FANK sequence. Then we hypothesized that by the generation of targeted deletions, we could increase the initial frequency of potentially therapeutic events. So in this case, we used the mutant-specific single-guide RNA together with a previously tested single guide RNA, by which we could generate a precise 18 base pair deletion that removed the premature stop codon and preserves the sequence of Fanconi gene. So using these two guide RNAs, we observed 5 days after electroporation that we were able to generate in a 12.5% the precise 18 base pair deletion out of a total of 18.3% potentially therapeutic indels that once again showed a marked proliferative advantage when cells were maintained in culture up to 60 days, in which we observed a 23% of the precise deletion out of a total of 35% potentially therapeutic indels. So using these two systems, the use of a single guide RNA and also the generation of targeted deletions, we confirmed the stable expression of the new edited Fanconi protein by Western blot before and after mitomycin C, which is an interstrand cross-linking agent to which Fanconi anemia cells are highly sensitive. And also, in order to see whether we could restore the Fanconi anemia pathway, we culture edited cells at increasing doses of mitomycin C, in which we observe the reversion of the mitomycin C hypersensitivity in these edited cells, in clear contrast to what observed in the non-edited Fanconi anemia cells, which confirm the restoration of the pathway and thus the reversion of the Fanconi anemia phenotype. So after demonstrating that the non-homologous enjoining-mediated gene editing could be a potential therapeutic strategy for Fanconi anemia, we wanted to evaluate the potential of this strategy to target other Fanconi anemia genes. So in this case, due to the simplicity of this approach, we designed a unique single guide RNA for the FANC-B, FANC-C, and FANC-D2 complementation groups. So in this case, we observed that gene editing was robustly efficient, reaching around an 80% of total editing events with an average rate of potentially therapeutic indels, which ranked from 21 to 31%. That also showed a marked proliferative advantage when cells were maintained up to 20 days in culture in the presence of mitomycin C. We also analyzed the reversion of the mitomycin C hypersensitivity in these cells, in which we observe a marked resistance to this drug in all edited cells, similarly as what observed in a healthy donor lymphoblastic cell line, confirming in all cases the correction of the Fanconi anemia phenotype. Moreover, a patient-derived lymphoblastic cell line harboring a heterozygous mutation in the BRCA2 gene was selected, since these cells are characterized by marked defects in homologous recombination. In this case, 5 days after electroporation, deep sequencing analysis revealed an 18.6% of potentially therapeutic indels that naturally increased over time when cells were maintained up to 30 days in culture in the absence of mitomycin C. Data that correlated with the marked resistance of edited cells to mitomycin C, similarly as what observed in a healthy donor lymphoblastic cell line, and confirming once again the correction of the Fanconi anemia phenotype. So even though the non-homologous enjoining-mediated approach is a really promising therapeutic strategy for Fanconi anemia, considering the deleterious effects of generating double-strand breaks in the genome, we started pursuing safer genome editing tools, such as the recently described base and prime editing strategies, which allow the generation of precise modifications in the genome without affecting DNA integrity. These systems do not require the use of donor templates. They do not rely on homologous recombination, which is quite important, at least in the case of Fanconi anemia cells. And also it is applicable in, or can be applied in non-dividing cells, such as hematopoietic stem cells. So focusing on base editing, base editors allow the generation of up to 4 possible transition mutations, which are constituted by a Cas-nickase fused to a single-stranded DNA, cytidine, or adenosine deaminase enzyme that allows the direct conversion of a C to T or A to G, respectively. These base editors allowed the precise conversion of the C or A located at a precise editing window, which is located at the 4th to 8th PAM distal nucleotides. We have also to consider that if an additional C or A is present within this editing window, this non-target base can become edited, promoting what is known as a bystander edit that reduces the product purity of the system. Also, base editors are characterized by two types of off-target activity, those that depend on the intrinsic affinity of the Cas domain, known as single guide RNA dependent, and those that rely on the affinity of the deaminase to bind to single-stranded DNA, that are known as single guide RNA independent. But apart from this, off-target activity is quite low with a characteristic reduced indel formation. And what's more important, in the case of Fanconi anemia, we could apply this strategy to target up to 40% of Fanconi anemia single nucleotide variants. Very briefly, the base editing strategy is driven to a certain place in the genome by a specific single guide RNA, where targets C or A are recognized and deaminated, generating an uracil, in the case of using a cytosine base editing strategy, or to an inosine when using an adenosine deaminase approach. Then the simultaneous nick in the non-edited strand containing G or T bases allows the resolution of the recently formed heteroduplex, permanently introducing the desired edit in the genome. So in collaboration with Jacob's Korn lab from the ATH University, in order to see whether adenine base editing was a possible therapeutic strategy for Fanconi anemia, we conducted first experiments in order to see whether we could apply this approach to target hematopoietic stem cells. So with this purpose, first set of experiments were conducted in healthy donor cord blood or mobilized peripheral blood CD34 positive cells, in which two different adenine base editors were tested, the ABE-MAX and the improved architecture known as the ABE8E base editors. These two constructs were used in combination with a specific single guide RNA targeting the AAVS1 safe harbor locus that allowed to study the behavior of these cells over time without causing any detrimental effect on targeted cells. And then, in order to study the maintenance of the repopulating ability of hematopoietic stem cells after adenine base editing, edited or mock cells were transplanted into immunodeficient NSG mice. And then monthly post-infusion, these cells were collected for engraftment and editing analysis. So what we observed was that we could reach similar and high levels of human engraftment in primary recipients that had been transplanted with ABE-MAX or ABE8E edited cells, similarly as what observed in the non-edited cells. Also, we didn't observe any significant differences among the different hematopoietic lineages evaluated within the edited or mock cells that had been transplanted in these primary recipients. So then we confirm that adenine base editing can efficiently target long-term repopulating hematopoietic stem cells without affecting their engraftment or repopulating ability capacity, as observed in secondary recipients, and in secondary recipients in which we didn't observe significant differences, once again, within the edited cells and the non-edited ones. Then, remarkably, when the editing efficiencies were analyzed in long-term repopulating hematopoietic stem cells from primary recipients, we observed high levels of precise A to G base conversion before and after transplant in the ABE8E edited cells, reaching around 60 up to 60% in primary recipients 90 days after transplant. Levels that were maintained in secondary recipients analyzed 90 days after transplant, which confirm or support that long-term repopulating hematopoietic stem cells can be efficiently edited by adenine base editing. So once proven that adenine base editing was a feasible strategy for hematopoietic stem cells, we aim at evaluating the therapeutic potential of this approach. So with this purpose, lineage negative or CD34 positive enriched cells from 3 different Fanconi anemia patients harboring the previously described stop codon mutations were selected. And then, due to the good results observed with the ABE8E construct, we used this one together with a specific single guide RNA by which we could directly revert the nonsense mutation into a therapeutic missense mutation. So we observed that the intended A to G base conversion was highly efficient, as we can see here in this graph, analyzed 5 days after electroporation in liquid culture, in which we obtain up to from 40 to 60% of the intended A to G base conversion in these different Fanconi anemia H species. And also, we observed that this intended conversion showed a marked proliferative advantage when we analyzed hematopoietic colonies only 14 days after electroporation, where values close to 100% were observed in these colonies without observing any bystander modification, probably due to the proliferative advantage of colonies only harboring the precise modification. Also, since Fanconi anemia cells are highly sensitive to mitomycin C, as previously shown, we analyzed the survival in these colonies to mitomycin C, in which we observe a marked resistance of these cells to the drug, in contrast to what observing the non-edited Fanconi anemia cells, which confirm the reversion of the Fanconi anemia phenotype. Also, in order to analyze the safety of adenine base editing in Fanconi anemia hematopoietic stem and progenitor cells, we analyzed the top in silico predictor of target sites for the specific single guide RNA in patient 2 and 3, in which we didn't detect any elevated A to G base conversion in the different genomic sites analyzed. So after showing that base editing was a really efficient strategy for Fanconi anemia, we wanted to evaluate a different, also safe gene editing tool that could be applied to target higher number of Fanconi anemia related mutations. So we moved to prime editing. That is a strategy that allows the generation of targeted insertions, all deletions, and up to 6 possible base pair conversions. So prime editors are constituted by a Cas9 case fused to a reverse transcriptase that is driven to a certain place in the genome by a specific prime editing guide RNA formed by a protospacer, an RT template that carries the desired edit and the primer binding site, where it hybridizes to the targeted region and starts the reverse transcription. So it's a very versatile and flexible strategy, and also with a characteristic low of target activity and a reduced indel formation. And in the case of Fanconi anemia, we can apply this strategy to correct up to 65% of Fanconi anemia variants. So very briefly, as just mentioned, the prime editor is driven to a certain place in the genome by this specific prime editing guide RNA that binds to the target region through the protospacer from the PEG RNA, allowing the generation of a nick in the opposite strand and the hybridization of the primer binding site to the nick strand, then allowing the reverse transcription using the RT template that is present in the prime editing guide RNA, permanently introducing this modification in the genome. So up to now, different prime editing systems have been described. First of all, the P2 and P3 systems were described, which consisted on the use of the prime editor and also the PEG RNA, known as P2 system, and then an improved version in order to enhance the editing efficiency, in which required the use of an additional single guide RNA to target the non-edited strand, and then use the edited one as a template to introduce in a higher frequency the specific modification. Then different studies shown that certain mismatch repair genes inhibit prime editing efficiencies, so 2 novel prime editing systems were described, known as P2 and P5. They use the transient co-expression of a dominant negative mismatch repair protein, in this case the MLH1 dominant negative, together to the P2 system, defining what is known as P4 system, and then in the P3 system, giving rise to the P5 system, in which we could enhance prime editing events and reduce indel formation even more. So in order to see whether prime editing was feasible in Fanconi anemia, we selected the previously described stop codon mutation in which we tested in patient-derived lymphoblastic cell lines the P3 and P5 systems. So we used the P3 system together with a specific primary, the P3 and P5 system, together with a specific prime editing guide RNA, by which in this case we could directly convert the nonsense mutation into the wild-type amino acid. So we observed that both P3 and P5 systems were highly efficient in patient-derived lymphoblastic cell lines harboring this precise mutation, in which we observed 5 days after electroporation up to 40 and 20%, around 20% of the precise correct edit. We also observed that when these cells were maintained in culture up to 60 days, the precise modifications showed a marked enrichment over time, reaching around from 60 to 70% of the precise modification. Also, we analyzed single clones of these cells in order to see the nature of prime editing events in these cells, in which we observed that in most of the cases, the precise modification took place in both alleles in more than 50% of the cases. Then, in order to see whether prime editing was also a safe approach for Fanconi anemia, we analyzed the top in silico predictor of target sites from both the prime editing guide RNA and also the single guide RNA used in both P3 and P5 systems, in which we didn't detect any off-target activity among the different off-target sites analyzed. And then, in order to see whether we could restore the Fanconi anemia pathway, we analyzed the expression of the FANK protein and also FAND2 monoubiquitination, in which we observed that we could restore the expression of these both proteins in both P3 and P5 systems, and also to see the proper mobilization of the FAND2 protein to the DNA lesion. We analyzed foci formation, in which we observed that it was completely restored in both P3 and P5 edited cells, similarly as was observed in a healthy donor lymphoblastic cell line. And then we also cultured these cells to increasing doses of mitomycin C, in which we found that prime-edited cells showed a marked resistance to this drug, behaving as a healthy donor lymphoblastic cell line, in clear contrast to what observing the non-edited Fanconi anemia cells, which confirm in this case the correction of the Fanconi anemia phenotype and also correlated with the Western blot at foci formation analysis. So as a conclusion, overall, our results have demonstrated that the use of precise gene editing tools can be applied to specifically correct Fanconi anemia-related mutations, depending on their nature. So in one hand, we have observed that the non-homologous enjoining-mediated approach is a really efficient strategy in Fanconi anemia lymphoblastic cell lines that can be applied to correct different mutations from different Fanconi anemia complementation groups. Also, we have observed that it could be applied to specifically correct different framesheets or nonsense mutations, but in this case, in less than 40% of the Fanconi anemia-related variants. So then, in the case of base editing, we have observed that is an efficient strategy that edits hematopoietic stem cells from healthy donors, preserving their long-term repopulating capacity. Also, we have seen that it's applicable to point mutations, in our case, up to 40% of Fanconi anemia single nucleotide variants, and which is highly efficient to correct Fanconi anemia hematopoietic stem and progenitor cells, by which we could specifically revert a nonsense mutation to a missense therapeutic mutation. And then in the case of prime editing, we can say that this strategy is quite efficient in Fanconi anemia, by which we can revert a mutant sequence to the wild type. Also, it's a safe strategy that has a reduced undecided editing events, such as indels that are quite detrimental in this case, and also that can be applied to correct most of Fanconi anemia-related mutations, in our case, up to 65% of Fanconi anemia-described variants. So, of course, I would like to thank all this work to our group that is led by our principal investigator, Paula Rio, and the rest of the group, Laura, Lara, Bea, Irene, and Marta. Also the head of our unit, Juan Bueren, to our external collaboration, especially to Jacob's Corn Lab that is Herman Karasu, Sebastian, and Alexandra. Also Davis Liu lab, because they've helped us a lot with prime editing strategies. For sure, all the patients, families, and clinicians that constitutes the Fanconi Anemia family, and all of you for listening. Thank you.
Thank you very much, Laura. Can you show the last slide? Okay. So I just want to draw your attention that we have a next webinar already in 14 days by Silvia Roman, and she's going to talk a bit more about epigenome editing and its application in CAR T cells, a subject that is very important for conquering cancer, and is considered as the new medicine of the future for cancer therapy. I think you can stop sharing, Laura, and I will go over to a bit of questions. First of all, thank you very much for this clear overview. It's true that for these gene editing strategies, Fanconi anemia is a perfect target because of the fact that it gives a selective advantage to the cells once they are corrected. Nevertheless, I guess we both agree, or maybe you can comment a bit on that, that we would at least need some minimal correction in the patients for the reconstitution of the hematopoietic system not to take too long. Can you comment a little bit on this part?
Yeah, sure. It's true that in the case of Fanconi anemia, we lead with this advantage, the proliferative advantage of these cells. But for sure, we need. It is true that theoretically, if we have one cell corrected, we can reconstitute all the hematopoietic lineage. But in the case of a clinical application, we will need a basal percentage of editing. Also, this is related with the viability of these cells and so on. So that's why we are, in one hand, really focused on searching different safe editing tools, and also, in the other hand, quite efficient ones. So we are working a lot on optimization and working a lot with efficiency, because it will be the ideal. It will be to reach as much as possible of a good percentage of gene editing in these cells, for sure. So yeah, we have this proliferative advantage, but we also would like to start with a good percentage of editing in the pool, because, for example, for an ex vivo strategy.
Maybe also the reason for that might be to nevertheless stay polyclonal and not to oligoclonal in the case something might go wrong. It's always better to start with a polyclonal.
Yeah, of course. Mm-hmm.
That from gene editing strategies from before. Mm-hmm. So one of the things I was also wondering about is, you say that Fanconi anemia cells are doing easier non-homologous enjoining than healthy stem cells, blood stem cells. Can you estimate how many fold they are better, and why this is exactly? Is it known?
Well, I will say, it's true that the non-homologous enjoining repair mechanism is enhanced in Fanconi anemia cells specifically, but in the case of quiescent cells, it is also the preferential mechanism. So we have these two advantages, let's say, right? Because the homologous recombination, and we've seen this in many different strategies, is less efficient, because in stem cells, such as hematopoietic stem cells, this is not the first choice of the cell for repairing double strand breaks. So I don't know exactly how much in comparison. I mean, a healthy donor cell, how much is the non-homologous enjoining pathway versus the homologous recombination one. This choice, I don't know if it's not 50-50 for sure, but I mean, I don't know the percentage exactly. But in a healthy donor, if we talk about a healthy donor background, for sure the non-homologous enjoining will also be more efficient than the homologous recombination.
And one of the things that I found a bit not strange, but that struck me was that when you go not only for Func A, but for corrections in Func B, Func C, Func D, I had the impression that for some corrections in these repair genes, you had a better expansion or a faster expansion of the corrected cells. Is that right? Or is it related to the gene that is targeted?
I mean, I think it's more related with the viability of the cells, because we've seen maybe targeting same gene for somehow in the cell pool at earlier time points, the viability is less than in a different experiment, and they expand better in one experiment than another. But if you maintain even more the cells, they reach the same percentage of correction. But yeah, this might be related with the viability at earlier time points. It's true that it depends on the gene also, because some of the patient-derived lymphoblastic cell lines are more complicated depending on the affected gene. For example, the BRCA2 gene, it's more complicated than other ones. But in this case, I think it's more related with the viability of the pool, of the cell pool. I can hear you, else I think. Yeah.
And then I have another question in relation to, so all these components are introduced in the stem cells via electroporation of what we call RMPs. So the RNA fused to protein, and we electroporate it. This is a quite harsh method, especially knowing that Fanconi anemia cells are very fragile. Even if you stimulate them, they might die quite quickly. So it's very good news that even in the quiescent state, these mechanisms work very well for these cells. I agree. But do you think that there might be other delivery methods that might be a more given advantage for the cells, especially knowing that we have few of these FA stem cells, they are fragile, and so on and so on. And we need enough cells to reconstitute the patient in the end.
Mm-hmm. Yeah, for sure. For a clinical application, we will have to try to move out of the electroporation system. But in this regard, we are really working very hard on optimizing different delivery tools based on viral-like particles, also lipid nanoparticles, to avoid this process, right? Because in the case of Fanconi anemia, and also in the case of healthy donor hematopoietic stem cells, is a quite toxic system, right, for the cells. I mean, the viability is quite reduced after the electroporation. So we would like to move to another systems. And also the idea will be an in vivo strategy for sure, but for the ex vivo one, yeah, we are working really hard.
There we need a lot of things to come together, right? A good delivery system and a targeted delivery system, which will only transduce
specific one
stem cells and so on. Yeah.
Okay.
So then there's one other question I have. It's more just a question to you, because you are really into this for Fanconi anemia. If we put aside the efficacy, not the efficacy, but the number of mutations we can target, because it's clear that for prime editing, we have more possibility. What do you think would be today the best and the safest strategy for Fanconi anemia correction in hematopoietic stem cells? Base editing, prime editing?
So it really depends, I would say, but I think right now the best strategy for me will be base editing, because in contrast to non-homologous enjoining, even though we can target more or less, a little bit more with base editing, but more or less the same percentage of Fanconi anemia mutations, it's a more specific tool, and it's also a safer tool. and it's quite efficient. I mean, and it's been tested also in the clinic. So I will go with this one directly. Prime editing is a really sophisticated method, and it's like ideal to being able of reverting a mutant sequence to the wild type and the original amino acid. But for now, I think that we have to work a little bit more on this. I mean, in different applications, right, on prime editing, it's quite a new strategy, also base editing, but I will say that in terms of efficacy, safety, and clinical application, I will select the base editing strategy. We'll see in a couple of years. I cannot hear you else. Yes, now.
Yeah, because just for the public, since I'm coughing a bit, I'm
Yeah.
cutting my mic, but I thank you very much, Laura, for your beautiful presentation and for answering our questions today. I'm sure there will be a lot of viewers also afterwards, because as everybody knows, these webinars stay on our website and can be followed at any time, and they will be continuously there. You can also go back to the previous one and to the ones that follow. Thank you very much, and I wish you a lot of success with the continuation of your research.
Thank you,
Laura.
Thanks a lot.
Bye to everybody listening.
Bye.